Hi everyone, my name is Lauren Yost. I'll be your instructor for Microbiology Lab. I'm just going to run through the lab, how everything's set up, what you can expect when you get in here, and all like the safety stuff that you need to know whenever you do get to the lab. That way when you guys come here, it's not completely foreign and strange, and you kind of have an idea, what you need to do. So first things first, you should be coming into lab with your lab coat on. I don't care what color your lab coat is, it's there to protect you from the chemicals that we'll be working with, the bacterial cultures that we're working with, and protect your clothes and all that. So that's what that's for. If you brought your lab coat with you and you put it on in the lab, that's okay too. Just be sure that before you start doing anything, that lab coat is on. If you fail to have your lab coat one day, or I have no extras, I don't have very many extras that I can supply, and I'm not able to accommodate that, then you will have to take an absence for that day. So keep that in mind. All right, when you come to lab, do not bring any food or drink in here. If you have water bottles with you, as long as they stay in your bag and you do not take them out at all during the lab, it's fine, you can keep them in your bag. You're also welcome to keep drinks and even your bags out in the hallway. We just share the end of the hallway with physics people, and that's it. Like, nobody else should be down here, and we've never had any issues with that before. So do whatever works for you. So you'll be coming in your lab coat, no drinks with you, and you'll have your bag. Typically you guys will have your backpack, right? So you'll come up to your bench, to your spot, and you can just put your bag underneath the actual bench, whatever works for you, but you do want to keep it out of the way so that people don't trip on it. You're going to be walking around, your classmates are going to be walking around, and I'm going to be walking around. So be sure that your bag is secured completely under the bench. Nobody's going to trip on it. It's fine. So that's where your bag is going to be. The next thing that you're going to do when you get into microbiology lab, you have your lab coat on, you have your bag under the thing, you don't have your food and your drink, you're going to disinfect your bench. This is literally just like a typical disinfectant, but it should take care of most of the microbes that we work with. So just squirt it on, get yourself a paper towel from one of the dispensers around the room. We have several of them, and just wipe it down. I will say that we do not use gloves in this lab. If you want to, you are welcome to. Don't let me stop you, but they are optional. So keep that in mind as well. If you have some from like a physiology class or something, you're welcome to use those. All right, so we've wiped off our benches, and we're gonna do our experiments and everything like that. Most of our stuff is gonna be done using aseptic technique, and that's gonna involve using fire, basically. So what we will do is have whatever instrument we'll be using that day. Most of the time it's going to be an inoculation loop, which I know you can't see very well, but you guys will be very familiar with it throughout the course eventually. And you'll turn on your gas at this valve here. You can hear it coming out. And most of you who have had chemistry probably are familiar with this. This is a striker. On the striker should be a visible flint still in there. If you're not sure or you're having problems with it, I can come check it for you and see if your flint needs replacing. So then you will just put a little pressure back into the striker. Don't just click it. Put pressure into it. Nobody does it right, and that's okay. I'll help you figure it out. You'll make some of the sparks come, and then when they contact the gas, it should light it on fire. It might take you a couple tries to get it, but it's not that bad. So you can see this might be a hazard in the lab. Basically, as far as the fire goes, girls have to keep their hair tied back. So if you have long hair or you think it's going to fall out in front of you while you're working and be a danger of getting into the flame, any hair past the shoulders, I want you to keep it back in, either in a ponytail or tucked into your lab coat to keep it away. This isn't a joke. This is something that happens. So I want you guys to be safe when you're working with the fire. It is fire after all, like it really is. So, so you're working with your stuff and maybe you have some tubes. Contaminated disposables, which just means like plastic petri dishes, that's easy to dispose of, swabs that we might be using, stuff that would be trash and wouldn't be reusable. If they get contaminated with the bacterial culture of some kind, they're going to go into the biotrash. And the biotrash is biohazard trash, and it is over here in this corner, in this red trash can guy, and you step on the thing for it to open. There's an orange bag in there, so you know what you're looking at. And that's where you'll throw anything that is disposable that's contaminated. If you spill bacterial culture on your bench or on you, whatever, you let me know, first of all. Any lab accidents you need to be letting me know about, whether it's just spilling something or you get an injury of some kind. But you guys let me know about that, and they're actually pretty easy to clean up. You will use your disinfectant to kind of soak over the actual culture material and then wipe it up with your paper towels until you get it relatively dry. Those paper towels will go into the bio trash. Then, if you need to, I would recommend going back over after you get that initial cleanup off, just the same way, more disinfectant and then wipe it off so that it's dry. That second paper towel can go in the regular trash because it should be relatively clean by then. So that's how we would deal with contaminated things. We're going to be making quite a few slides, like doing stains on our bacterial cells and looking at them under our microscopes. Those slides, when you are done looking at them, or any broken glass at all, is going to go into the sharps container here. And it's a typical sharps container, like you would see in a hospital, but big. So just be sure you put your broken glass in there so nobody is going to get hurt. And there's all sorts of signage on a lot of these things to let you know where you should be putting what as well. You can always ask me if you get confused too. As far as completed culture tubes, whenever we grow some of our cultures up inside of a tube to like analyze the growth of your, bacteria or whatever it is. Okay, this is a good one. All right, so like one of these kinds of tubes. First of all, you want to be sure you lift them by the glass and never the lid. The lid is not a secure lid. If I were to tip this over, it will pour out every time. It's made to be like that so air can get into the tube. Do not tip it where the liquid's going to come out the top, okay? It'll make a mess. If you grab the tube out of the rack from the lid, be sure the next thing you do is move some fingers down to the glass to hold it, because otherwise the lids, some of them will just slip off and then the tube will fall to the table or the ground. So that's that. When you're done looking at these and you say, oh, this is positive and oh, this is negative, which we'll learn about how that's all going to work later. But once you're done with them, you will remove any tape that you use to label it as yours, remove that, and then put it over here on this cart. It says on the cart to be autoclaved. An autoclave is what we use. It uses heat, steam, and pressure to sterilize things. And so we're going to put any old cultures that we want to be sterilized onto this rack. So anything that you have used in your tubes or something that you've like messed up, you didn't do the culture right or something, then you'll put it on this rack and we will autoclave it, wash the tubes and the lids, and then reuse them. So be sure that those tubes, as long as they're not broken, get put on this cart here. And racks, there's more racks back here if you need them. All right, so I'm gonna go ahead and turn this fire off because I think our demonstration with that was pretty fine. Okay. Right, so tie back on here. So the other thing that you have to have in order to be safe in microbiology lab that is required, sort of like we had talked about with the lab coat, right? That's gonna be, you need closed-toed shoes. Closed toe, okay? You can have shorts or a skirt on, that is fine, like I do, but be sure that your shoes have a closed toe on them. They don't have to be quite so closed as my boot, but they do have to be covering most of that kind of vulnerable area of your foot if you were to drop something dangerous. That's just the idea to keep you safe. Again, if you do come in with footwear that is not acceptable, Crocs, by the way, are not acceptable because they have like obvious holes there. So don't wear Crocs, don't wear flip-flops, nothing like that, no sandals, okay? If you come in with that on and you forgot or whatever, I hope that you have extra shoes in your car because I will be sending you out of the lab and you will not be able to join us until you have appropriate footwear on for your safety. So you've been warned. Whenever we are working in the lab, we want to work with what's called aseptic technique. And we are going to learn about that in our first lab, actually. So you don't need to worry too much about the details yet. But Aseptic technique means when we're working with bacteria and we're trying to move it from tube A to tube B, we want to make sure that we're only moving it from tube A to tube B, and it is not getting anywhere else, not on you, not on your environment, not on any other tubes, and we want to keep it that way. We're going to learn techniques on how to, manage that on our first lab. But always use that. So if you come in and the assignment is, inoculate, put some bacteria onto this plate, don't just pour it on there, right? Always use aseptic technique, always. It'll become so second nature to you, you won't even think about it, because we're going to use aseptic technique every single day in the lab. So next thing, if I'm not in here, you guys shouldn't be working with fire, okay? If I need to grab something in the, storeroom or something that we ran out of, and I need to leave temporarily, I will usually check to be sure there's no Bunsen burners on. To me, that's the biggest concern if I am going to be leaving the lab. That's the most dangerous thing that could be happening there. So definitely if I'm in here, there will be no working with Bunsen burners. If you forgot a thing, you will wait until I return to relight your Bunsen burner and do that thing. So there's that. Don't drink the laboratory alcohol. We actually use quite a lot of alcohol in a lot of our labs, in our staining and everything like that. For example, when we are going to be doing our gram stains, which are one of our most basic of our stains, we will be rinsing with ethyl alcohol. Whether you can read or not doesn't really matter, but that's what this is. It's ethyl alcohol, also known as ethanol. This is drinking alcohol. This alcohol, for laboratory purposes, they have tinctured it with methanol, essentially, and that's wood alcohol. And if you don't know about what that is, it will make you go blind if you drink it. So do not drink the lab alcohol. It is, been treated with that on purpose to make it so you can't drink it, because otherwise, and I'm not kidding, otherwise we would have to have a liquor license. That's the truth. So don't drink any of the alcohol in the lab. If you have any immune system issues, a lowered immune system, if you are pregnant, that lowers your immune system, by the way, or taking things like Humira, that might make you more susceptible to illness, please let me know as early as you can in the lab so that we can make some accommodations for you and make sure that you're safe in the lab and not handling anything that could be an issue for you. The things we work with in the lab, for most healthy people, not a problem at all. That's why we don't even wear gloves. But if you have an immune system problem, I can't say for sure that you would be completely safe. So just let me know and we'll try to make some accommodations there. If you're not sure about anything that's going on in the lab, don't just wing it. Ask a question. I'm more than happy to ask questions. I've never ever gotten mad at anybody for asking questions about anything in my class. I want to be sure that you understand things and feel safe in the lab as much as possible. So please feel like you should be able to approach me. I'm here to help you have a pleasant experience in the lab, learn as much as you can, and be as safe as possible. Some other things that aren't going to be addressed on the lab safety document online, which is available on Canvas, are fire extinguishers. There's one back here. There's also another one at the opposite end of the corner. Just trust me that it's there. And then we have a first aid kit, one over here on the wall with the windows. You'll know. Okay, I want to just show it really quick, just so we know where it is. Oh, that's a good one. So we have this first aid kit, and we also have another one at my seat at the front where I usually sit by the computer. You'll know where that is. So if there is any sort of accident and you need to get into that, that's where they are, so that you're aware. The other thing is, when you come into lab, most of our exercises, I want you guys to read your lab manuals before you come to lab so you know what you're getting into. That's why you have to do your lab homework before you get here. If you haven't done your lab homework, you will not be allowed to participate in the lab. That's part of being prepared for the lab, so you're not wasting my time or your group mate's time. So basically, there are on the sides of the benches where you're at, there's little hooks, and on those hooks will be hung like a little guide for the day, okay? This is actually where your credit for the assignment for the day is gonna come from. So we have at the top, basic rundown of the concepts of how this works. Then maybe some important terms you might wanna know related to it. In the orange box, which is the part that you probably care about the most and probably don't care about the rest of it, this is the procedure. And I have made sure in bold and underline the basic, word associated with that step. I will have these, each student will have their own of this. I'll also have a version of the procedure put onto the TVs at each end of the room, so you can look at that for your ease as well. And that way your lab manuals aren't getting gross when they're being exposed to spills or anything like that. I wanted to be sure that you guys didn't have that issue, and you can take them home, your lab manuals anyways, take them home for studying, and that'll be your study portion. And you can use that as well for the homework. So you come in and you do your procedure. You'll notice that there's a back to it as well. So, so yes, and then it's just like what it looks like. There are questions associated with this. It's basic questions about what this exercise did today. And then at the bottom for most of the exercises, I will have a grand rounds question where you will go around and get some results from some of the other tables. They'll help you interact with your fellow classmates, but also, which is something I feel like doing a grand rounds thing very similar to what you would be doing when you go to nursing school and actually start learning in that environment, in a hospital environment. So that's what I want to expose you guys to. Anyways, and I'll also let you guys look at different results from what you guys saw. That way, when we get to the test questions, you kind of know that what you're looking at is what you expect to look at. So it should make sense at that point. But yeah, so then at the end of that, this is, I don't know, the best part, I guess, sort of. You go through and you answer those. Basically, it just shows me that you understood you don't have to get super, super picky about your answer, I'm just saying. But show me that you understood what was going on in the procedure. In every drawer is a small fine tip dry erase marker. This is what you will use to write on the little handout. And you will just go in and you'll answer as best you can. And then, whatever, write your answers to your questions that are appropriate. They don't have to be in full sentences or anything like that, just so that I can understand that you understood what you did today. And then take a picture of it with your cell phone. And then you will submit that online in the assignment associated with whatever this says at the top of it. So that way, you can just turn it in like that, and we don't have to carry any nasty papers back and forth or whatever. You'll just put that online and submit it. There is a deadline. The deadline for submitting your actual participation pictures will be before the next lab exam. So for the lab midterm and for the lab final, those are the deadlines for those. Okay, and then after you take your picture, you will kindly either erase with your dry erase or clean it off with your disinfectant. Probably disinfectant is the best way to go. Before you put it on the side for the next lab to come in. You may not be the only lab for the day. So for the very last thing for your day, well, there's two things, but at the end of the day, you'll do another disinfecting. So you come in at the beginning of the day, you're going to assume the people before you didn't clean. Just assume everything is contaminated all the time. That's probably the best way to work. And then at the end, you're going to clean up because you're a good student and you don't want to leave a mess behind for the next people, right? Even though they're going to assume you were dirty too. So, so that's the idea with that. Again, regular trash is fine for that paper towel. And then you're going to come over to the sink. There's a hand washing sink here, as well as in the back where my computer is. You can use either one. If it starts getting backed up, people end up waiting in line. Remember that you should be removing any jewelry that you might have whenever you go to wash your hands and follow proper hand washing procedure that you should have learned online for the lab exercise before you came in for the first time. So that is, yeah, you wash your hands, and that is that. And yeah, so if you guys have any questions about that, let me know, email me, leave comments, whatever works best for you. And I'll see you guys at the first lab. Can't wait to meet you all.